a lot of cloning in this last week or so, and over the course of my nearly 10 years in biochemistry, I've made nearly hundreds of plasmids, and so I kind of, in order to keep track of them all, I've developed this sort of system, and so I thought I'd just share with you what works for me. This includes things like keeping a detailed plasmid inventory with information about all the different constructs. It includes keeping my cloning intermediates, so before they're verified sequences, separated physically from my actual plasmids that I have sequenced and know are good. This includes things like making glycerol stocks and all of this sort of thing. So here's just a quick look. So first, starting with that plasmid inventory, I've done a post on this before, and I have a template that you can go and download and use for yourself. Basically, this has information about the plasmid. This includes antibiotic resistance, so you make sure that you plate it on the right plates. Plasmid size, insert size, if it's for protein expression, what's the promoter, what's the size of the protein and the PI of the protein, and all this various stuff. And so having it all in that one spreadsheet makes things really, really handy. You'll also see, if you look at the template, that I give my plasmids a number. This makes it easier to basically not have to write as much on a tiny little tube and then be wondering, is that a C or a D, and that sort of thing. Also, make sure you put the date on all your tubes. That's going to make it helpful for cross-referencing as well, and I have a whole post on the importance of keeping a date. So that's the plasmid inventory, and it's going to come in handy throughout the entire lifetime that you're using this plasmid. But what's also going to come in handy is keeping track of things when you're making the plasmid. And so in the course of cloning and mutagenesis and all this stuff, you're going to make a lot of duds. No offense, nothing personal, it happens to all of us. And so, for example, this is why you always want to send your plasmids for sequencing, even if you've done something like colony PCR or analytical restriction digest, so you think they're probably okay, send them for sequencing to verify. So, for example, I recently did 25 mini preps on Monday because I was sending 5 different colonies for 5 different constructs, and it was a time crunch, so I'm like, I can't redo this, I need one of these to work, and then almost all of them worked, and I wasn't sure whether to be happy that they all worked, or to be sad that I wasted all that time on ones that didn't work, on things that were working because I thought they might not work. But anyway, most of them worked, but there were a few that didn't. And so it was a good thing that I'd done that sequencing. And it was a good thing that I kept them physically separate from my verified plasmids so that I don't then go and accidentally use one of those duds. So instead, when I get the sequencing results back, then I go to my box of my cloning in progress box, and I find those tubes that were the bad ones, and I toss them. This way, I don't have to worry about then accidentally using them in the future. Now for the tubes that were good, now I'm going to put some more detailed information on the tubes. I kind of be lazy earlier on and just use little short codes like 14A, 14B, 14C, and then have notes on what those various things are and their concentrations. And then once I've verified them, I go and I write all that information on the tube so that the person in the future, me or someone else, is then able to, though, again, identify what that plasmid is, what the concentration is, all of this various stuff. And it'll go into my plasmids box, organized by their construct number and things like this. So everything is super duper organized. So I have those clones, and now I know that they're good. But for some of them, I know I'm going to need a lot of this plasmid. I'm going to want it in the future as well. So what I'm going to do is I'm actually going to take that plasmid that I was mini prepped out, so I purified it out of the bacteria. Now I'm going to stick it right back in those bacteria. And this time I'm going to plate it again. I'm going to pick some colonies. I'm going to do an overnight culture. And I'm going to do another mini prep as well as take a mil of that culture. So I'll do a 5 mil overnight tonight. I'm going to take a mil of it and make a couple glycerol stocks of it. So basically take the bacteria and freeze them with some glycerol so that they'll be stable long term in the -80 freezer. And then if I want to use them in the future, I can skip the transformation step and go straight from them being in the cells. And I'll take the rest of the culture and I'll mini prep them, so I'll purify more of the pure plasmid out of it. Now for some of these, for protein expression constructs, I'm actually going to need to transform them into a different cell type. And so, for example, these are in cloning cells, which are great if you want lots and lots of copies of the plasmid, but not great if you want lots and lots of the protein to be made. So instead, I'm going to have to transform them into cells like BL21, DE3, which have things like the inducible. If I'm using inducible expression with IPTG, then this will express the T7 polymerase, and much more on that in other posts. But basically, there are some bacteria that are good for making lots of copies of the plasmid. We often call these cloning cells. They include things like DH5 alpha. And then there are bacteria that are really good at making protein, things like BL21, these like expression strains. And so if you want to express the protein, now you're going to have to stick that plasmid into those other cells. And when you do that, then you're going to plate them and all that stuff again, and then you might want to make a glycerol stock of those. But typically you're not going to mini prep out of those. You'll get more colonies if you'll get more copies of the plasmid, higher concentrations if you do it out of one of those cloning cells like DH5 alpha. Okay, so you've done all that. You've kept track of your plasmids. Make sure on your plates too. So if you have cloning plates that were for like your cloning in progress, make sure you don't mix those up with the plates that you have for your verified sequences. So if you validated it, so basically on my plates that I'm doing for cloning, I make sure and I put on the plate that it's for cloning. So I use this strategy called SLIC, site and ligation independent cloning, and I have a post on that as well. But basically I'll write SLIC on the plate, and that way I don't go and accidentally take one of those colonies thinking it's one of my verified sequence colonies when really it wasn't. So usually once I verify the sequence that first time after the cloning, on the next plate after I've just done the transformation into the cells and plated them again, I don't sequence again at that point. I might later on sequence the plasmid, especially if I'm having some problems with it, but typically I could just go back to that plate within a couple months or so, and if it's longer term, then I'll go to that glycerol stock, or I'll take that purified plasmid and transform more of it and make more. But it's always nice to have that backup as the glycerol stock, especially in case like you accidentally use the last of your plasmid thinking there's another tube and there isn't, or something like this. So just a note when you're doing that transformation, when you have the transformation from a purified plasmid from like a mini prep, you don't want to plate the whole thing. You typically only plate like 50 to 100 microliters or so, whereas with the SLIC or whatever, you're going to have to plate a lot more because it's going to be less efficient when you're doing the cloning process. But when you have a purified, nice little supercoiled plasmid from your mini prep, the bacteria are going to take that right up, and so you don't need to plate very much. Just a technical note, so you don't like overload your plates. So yeah, so that's the basics. So keep your plasmid inventory, keep your constructs nicely organized. Try numbering if that works for you. Keep your in progress separated from your sequence verified. Make sure you do sequence verified, and then you toss anything that is sequence unverified or like sequence bad verified that you know is a dud. You don't want to use that in the future. Make more plasmid if you want. Make glycerol stocks if you want. Transform into expression cells if you want. And make sure that, yeah, you typically keep the plasmids in the -20, keep your glycerol stocks in the -80, and all should be good. And you can go and you can do all sorts of stuff. You can express proteins, you can mutagenize those, make more constructs, have more clones in progress, and then more verified clones, all that good stuff. And keeping organized is going to make your life a lot easier. So hope this helps. This is just how I strategized. You need to find what works for you, but start early on so that you don't end up with a bunch of things and then like have no clue what's what.